Hello, I have to do another story report for Mr. Wong's class. It is something to do with respiration and glucose and gas laws. All I know is that I need an A, so I can go to college and make a lot of money and my parents will brag about me to everyone. I do not know why this is important to me, but it is. So, these things are called respirometers, but they are really just test tubes with rubber stoppers that fit into the opening. However, you should notice that there are tubes sticking out of the rubber stoppers, and Mr. Wong said that we will be putting living things into the tubes. I think this is pretty cruel, so I might call the Humane Society and tell them about it, but wait, if Mr. Wong goes to jail, I might not get an A, so I think I will not call.
I am now a dude. Anyway, here are some things that we could put into our respirometers. There are germinating pea seeds and glass beads. If the seeds are germinating, that means that cell division is happening, which means that cellular respiration is happening, which means that glucose is burning, which means that oxygen is being consumed, which means that I can calculate the amount of glucose being burned by watching how much oxygen is being consumed, and you might notice that glass beads take up the same volume as the seeds. I think the glass beads are used. It works like this. A tiny drop of water is placed in the pipette, and as oxygen is being consumed, a vacuum is created in the pipette and the drop of water will be pulled more into the tube, and I can actually measure the volume being consumed. I also put absorbent cotton soaked in potassium hydroxide into the tube, which will react with the carbon dioxide to form a solid potassium carbonate. This is fantastic because now I can measure the amount of oxygen being consumed without having to factor in the carbon dioxide, but I still need to account for changes in temperature and pressure because I am dealing with a gas, but this is what the glass beads are for. I can set up a control with the glass beads to factor in temperature and pressure changes. I am so excited. Stopped us before we had a chance to get started and gave us these super cool wireless carbon dioxide sensors. Now we could measure the rate of cellular respiration by just looking at the amount of carbon dioxide produced. This will be a heck of a lot easier than the other stupid respirometers. No more pipettes. No more absorbent cotton soaked in potassium hydroxide. No more drops of water.
I decided to become a girl again. I am not sure why, but I think it is because girls are smarter than boys and I really need an A anyway. With the wireless carbon dioxide sensors, we could now concentrate on factors affecting cellular respiration instead of the stupid construction of that rinky-dink respirometer described in the AP Biology Lab manual published by the stupid College Bordeaux. I am sorry. I got off track for a moment. Anyway, with the carbon dioxide sensor we still needed to convert parts per million into an actual number of molecules. Mr. Wong attempted to explain this to us, but I have ADD and my listening skills. I will use these mealworms for my experiment. I think respiration rate will increase when they compete for food, so I will put 3 worms in the jar and measure their respiration rate. Next, I will put 3 worms into the jar along with some food and I bet their respiration rate will increase. I am excited to start this experiment next week, but I wonder how Mr. Wong will grade us. I need to remember to bring Mr. Wong a Diet Coke. Thank you for listening.